A little bit about his past. Dr. Loncaric is from Croatia originally, but he lives in Vienna. He received his undergraduate degrees and doctorate in veterinary medicine at the University of Zagreb in Croatia. And then he subsequently moved to Vienna, where he is a professor and works in the Division of Microbiology in the School of Veterinary Medicine there. He is an expert on the presence of antibiotic resistance bacteria throughout wildlife. He has published extensively in very prestigious journals, including Nature, which is about as high as you can get. And that's actually where I've seen some of his articles that are really very, very interesting. I'm sure he will make some references to that. In part of our pursuit to engage with One Health projects, where we look at the importance of microbial and biosystems in both animals and humans, we are really pleased to be working with Dr. Loncaric. He spent some time in Rwanda, which he'll talk about in his lecture. And I am happy to report that he is going to go with us on our next trip to Tanzania to help set up a project very similar in Tanzania in the Serengeti and Ngorongoro. As many of you know, I'm an infectious disease specialist, and one of the great problems in infectious disease, and I know Dr. Dichter's on the call tonight too, is the presence of antibiotic-resistant bacteria. It's a challenge that we have all over the world. In fact, more people in sub-Saharan Africa die from antibiotic-resistant bacteria than they do from HIV now. So it's a huge problem, and we don't really understand all of the dynamics of antibiotic-resistant bacteria. So understanding the presence of these microbes in other animal systems is critically important, and that's exactly what Dr. Loncaric does and will continue to do. So without further ado, I'd like to introduce Dr. Igor Loncaric, who is broadcasting live from Vienna. He had to get up. What time is it there, Igor? 4 o'clock, 5 o'clock?
It is 4
o'clock in the morning. Don't wake up your neighbors. Okay, I'll turn it over to you.
Okay, thank you very much. And good evening or good morning to everyone. And this is a pleasure that I can give this presentation. And thank you very much for inviting me. And this morning, I will speak about the antibiotic-resistant bacteria associated with wildlife. And this is the content of my presentation. First of all, I will speak short about myself, and then I will tell you how came I from classical clinical microbiology to my wildlife work, and then I will have an introduction where I will speak about resistance in bacteria, but that's just a general overview. And then I will present a couple of studies, and on the end we can discuss about my presentation. This information you got already, but just a short repetition. I studied veterinary medicine in Zagreb, Croatia, and then I went together with my wife. She's also vet, to do doctoral study at University of Veterinary Medicine in Austria. I did my the doctoral thesis at Institute of Microbiology, and afterwards I was for a couple of years something like postdoc at the Austrian Agency for Health and Food Safety, also here in Vienna. And since 2010, I'm at the institute. I come back to the university, and since then I'm working at the Institute of Microbiology at the University of Veterinary Medicine in Vienna. And in 2020, I did my habilitation and earned the title private docent. This is the interesting story with private docent because every time when I'm in the United States, the people, the students and colleagues ask me what does it mean, and I just put here. I quoted the Wikipedia here, and private docent is an academic title conferred at some European university, mainly German-speaking, in German-speaking country, to someone who holds a certain formal qualification that donate an ability and permission to teach and designate a designated subject at the highest level. And this is comparable with associated professor in United States, but it is not associated professor. We have some differences between private docent and associate professor, but it is similar. And then just a couple of typical European story. Just a couple of words about my university. The University of Veterinary Medicine was founded in 18th century. This is the third oldest veterinary school in the world, but the oldest veterinary school in German-speaking country. And on the beginning, everything was influenced by military interests and the field of human medicine. And now, what are my interests? The majority of my time, I'm doing clinical microbiology. What is a clinical microbiology? This is for those people who are not so familiar with how laboratories work, especially microbiology. This is just a simple example. If you go to the medical doctor will take you a sample, swab or something like that. They will send this to the laboratory, and then we are in the laboratory who are dealing with bacteria or with also fungi. The other colleagues, they are doing virology, etc., etc. And my research focus are on resistant bacteria associated with companion animals, like dogs and cats and small mammals. Why? Because we have a huge clinic, and on daily basis we are getting samples, and from these samples I'm generating also my generating interesting bacteria for my research. And then I'm also interested. My interest is also in resistant bacteria associated with livestock. And the last three, they are somehow connected with my presentation today. This is a resistant bacteria associated with wild animals, and bacterial diversity from domestic and wild animals are originated from different countries. And finally, I'm helping in improving veterinary biological diagnostics in developing country. I'm active since a couple of years in Rwanda. This is an Eastern African country. I will come back to Rwanda later during my presentation. And how the story with my wildlife work began. This was on the beginning of my so-called return to the university. The German colleagues, they have a project about European hare. You see here this very nice animal. What is the problem with European hare? The population of European hare is declining in the last couple of decades, and nobody knows exactly why is this so. And then they organize a project, and the German island of Pellworm. This is here, and then every year we got approximately 40 to 50 fecal samples and the nasal swabs of these animals, and then we start to investigate. And I think this was in 2011. This was probably a couple of months after I came to back to the university. I got a swab from this hare, and then I isolate Staphylococcus aureus, and then we perform susceptibility testing, and this susceptibility testing told us that this bacteria is methicillin resistant, meaning that we have methicillin resistant Staphylococcus aureus, or MRSA. And then I was so happy, and then because in this moment I didn't know that something new is here, but then I took a look on PubMed. This is the biggest database with medical publication, and then I couldn't find any connection between European hare and MRSA. And then I told the colleagues in the laboratory, super, this will be publication, and this is new and things like that. And they told me, yeah, Igor, you are crazy. But on the end, we got a very good, we got a very nice publication. It was and until the present, a couple of times, I think approximately 100 times it is cited referent until the present. And what is the much more interesting in this situation, that our MRSA was not MAC-A, which is the majority of MRSA, they carry MAC-A again, but we had MAC-C, which is which in those days it was a quite new information. And then if you take a look today, for instance, European hare and MRSA, probably you will find only 5 or 6 publications, and 3 are associated with our working group. So very interesting situation. And then we started to think what is the reason why this European hare has MRSA. But you know, we found just MRSA either in in nasal swabs or in somewhere in wound or something like that. But in general, they were not sick of MRSA. Until present, we don't know which role play MRSA in this animal, but it was interesting for us why this animal has MRSA. And then we take a look on this island, and you see on this island you have a lot of livestock running around, and they share the same habitat with hare. And then we organized the second study. The German colleagues sent us nasal swabs of sheep and cattle, and what we found that that sheep and cattle have the same, in those days, the same MAC-C MRSA like the hare. So somewhere in the past, the transmission, interspecies transmission between wildlife and wild animals, in the case hare, and livestock happened. So and they're genetically ident with the applied methods in those days. In those days, we didn't have full genome sequencing, but everything showed us that they are either the same or the very close connected, this MRSA from hare and livestock. And then I come back to the resistance. Just a couple of information for those people who are not so familiar with antimicrobial resistance. What is antimicrobial resistance? This is non-susceptibility of bacteria to antibiotics. I strongly believe that everyone knows what antibiotics are, and the level of non-susceptibility is measurable as a minimal inhibition concentration and depends on tested antibiotics, tested bacteria, and the resistance mechanism present in tested bacteria. And in general, we distinguish between intrinsic resistance, which is species or genus specific. For instance, genus-specific intrinsic resistance is the resistance on cell-free mycoplasma, meaning all antibiotics like beta-lactam antibiotics, the most important class of antibiotics, they interfere with cell wall. So if bacteria doesn't, if one bacterium doesn't have cell wall, this antibiotic cannot work. And then we have acquired resistance, which is extremely important. This is mainly through strain. It is mainly strain specific, either through mutation or acquisition of resistant genes. And if we are speaking about resistance, the other term is also important. This is a multi-drug resistance, and this is in veterinary medicine. The resistance, if you are performing susceptibility testing, if one bacteria is resistant or 3 or more antibiotics from different antibiotic classes. For instance, if we have Staphylococcus aureus resistant to to tetracycline, or which is tetracycline, erythromycin, which is a macrolide, or chloramphenicol, or other antibiotics that mean this bacteria is multi-drug resistant. If we speak about MRSA, this MR, this is for methicillin resistant, but not for multi-drug resistant, because you can have methicillin, you can have MRSA, which is not multi-drug resistant, and you can have Staphylococcus aureus, which is not methicillin resistant, but multi-drug resistant. So it's a little bit complicated. And I took this picture from World Health Organization, and these are causes of antibiotic resistance. You can see that on the first place is overprescribing of antibiotics. Then you have problems that the people are not finishing with their treatment. Then what we as a veterinarian always have discussion with, especially with human, with medical doctors, is that in the veterinary medicine too much antibiotics are used, and then we have poor infection control in hospitals and clinics. And in my opinion, one of the most important issues is hygiene. And on the end, it is the lack of new antibiotics. So the development of new antibiotics is much slower than the development of resistance in bacteria, excuse me. And then I have another picture from World Health Organization. This is how antibiotic resistance spread. Just a short couple of words about this. It is actually everything interconnected, beginning from animals to hospitals, and on the end, the people can be affected with this kind of problem, with this antibiotic resistance. The environment play also an important role in the spread of antibiotic resistance. And I found this very important sentence about antimicrobial resistance, and they say from the people in World Health Organizations say that antimicrobial resistance threatens the very core of modern medicine and the sustainability of an effective global public health response to the enduring threat from infectious diseases. And from CDC in United States, the antibiotic resistance is one of the biggest public health challenges of our time. And then why is actually antibiotic multi-drug or drug resistance so important? Because, like I told you, it is one of the biggest challenges for the global health, can affect anyone. Recently, I was in hospital, and if you are a microbiologist and working with multi-drug resistant bacteria, and if you are in hospital and hear this person in 2 rooms on the left or 2 rooms on the right has, I don't know, ESBL, a second extended spectrum beta-lactamase producing enterobacteria, or you have someone with MRSA, you are alert very much, especially as a microbiologist, because we are seeing bacteria everywhere. And antimicrobial resistance occurs naturally, but misuse or suboptimal use of antibiotics is accelerating this process, and a growing number of infections are becoming harder to treat as the antibiotics used to treat them become less effective, and higher medical costs and increased mortality we have. These are a couple of information. Take a look. In 2019, more than 1.2 million people globally die on antimicrobial resistance in 2019. And so we come. I will come to my first study that I would like to present you. This is about beta-lactamase producing Enterobacteria and MRSA from rooks. You see, this is this bird. It's actually very smart and very nice bird. And what was objective of our study in those days? We wanted to compare two population of these rooks, one migrating and one resident population, and we took the fecal samples of these animals and perform selective bacterial isolation on presence of broad-spectrum beta-lactam resistant Enterobacteria and MRSA. And we wanted to know if this animal, rooks, could be potential reservoir for this bacteria. This is the map. Here is the Vienna, and in the eastern part of Vienna, we have this Lobau. This is a national park. It is also a recreation area, especially during the summer times you can go there and walk, and it is a very, very nice place. And from this place, we took the fecal sample droppings actually from a migratory population, and the 50 or 60 kilometers south of this was our residence population. 50 to 60 kilometers is approximately 35 miles south from Vienna. Is this red? What is this? This red dot is our resident population. Why I call this resident and migratory population? Because in the late October, usually the rooks are coming from eastern part of Europe. We have this population in Vienna, which is mainly from Ural region. This is approximately between Europe and Asia in the Russian Federation, but also from Belarus and the eastern part of Poland. They are flying to be here in Vienna, not only in Vienna, in Austria too, and in other parts of Europe to be during the winter times, because the winter is not so hard like in this eastern part of Europe. And then they're flying to Vienna and stay here until the end of March or April, and then they're flying back. So we wanted to see if the native population, which doesn't migrate, which kind of resistant bacteria this resident population has versus migratory population. And what we did, we took 54 samples of migratory population, fecal samples, and 102 of resident population. We performed then isolation and identification and characterization of bacteria, and performed a statistic analysis. And what we found on the beginning, I was actually shocked how much ESBL, this extended spectrum beta-lactamase producing Enterobacteria, and AmpC, this is a cephalosporinase, are present in migratory population. But also, for the first time in rooks in general, we found MRSA, but only in 5 out of 54 samples. And our resident population, we found also ESBL, but 3 out of 102. And what was interesting, that the proportion of antibiotic resistance is significantly higher in the migrating than in resident population. And this is especially with beta-lactam antibiotics and trimethoprim sulfametaxazole. And what is the most important finding of our study is that we found in one E. coli, which is multi-drug resistant. It is human-associated and pandemic, and this is the sequence type 131. And what is also interesting that these 5 MRSA which we found are pantone-valentin leukocidin toxin positive, which is they are also human associated and belongs to clonal complexes 1 and clonal complexes 22. Sorry, I forgot to put 2 more. It should be actually 22. So these are typical human-associated clonal lineages of MRSA. So very interesting situation. Why? Because, you know, if you are going to the national park to do recreation, and then with your child or I don't know, with your animal, they are allowed to go there with the dogs, and you have droppings with this kind of bacteria. This could be a public health issue. And why we choose rooks? Because they have a gregarious lifestyle. Also, if you take a look especially in Vienna on dog zones, I don't know what is the name in America. I think it is dog parks and something like that. We have here in Vienna a lot of these dog zones where you have a population of this urban wildlife like rooks. They share the same habitat with dogs and people. And you know, if the dogs are going around and then they go home and share the same habitat with their owners, it could come to the transmission of different microorganisms, including multi-drug-resistant bacteria. And rooks could be indicator species, especially in urban wildlife. And what can they tell us? They can tell us about environmental contamination with antibiotic-resistant bacteria. And also we have other animals in urban wildlife like rats and in pigeons, which do the same. We also publish a study with rats and found also very interesting bacteria, also multi-drug resistant. And the pigeons, on the beginning of my study here at the university, I did a couple of small studies with pigeons, but I never published this. But we have also ESBL, this multi-drug resistant Enterobacteria, also in pigeon, in rats anyhow. And then I switched the continent. I go from Europe to United States to North America. And I would like to present the study which I did with my colleagues from Western University. There in California at the Mono Lake, and we characterized and selected bacteria isolated from cloaca of nestling ospreys. And what was the objective of our study is to study the presence of enterobacteria or enterobacterales, enterococci, staphylococci, mammalicocci in cloaca of nestling. Why this bacteria and not other? Because these bacteria, enterobacteria, enterococci, staphylococci, and mammalicocci are known to be antimicrobial, to have acquired antimicrobial resistance. That's why this was important for us. And also the other objective, the second objective was may osprey be good biosentinels for the presence of antimicrobial resistant bacteria in the environment. And for those people who, I strongly believe that everyone knows where Mono Lake is. This is in eastern part of Central California. It is a very nice place. Take a look on these pictures. I got this from Professor Miguel from the Western University. This is a very good colleague of mine. This is so-called tufas. This is a calcium carbonate structure that appear after the water is the level of water is always smaller and smaller, and then these structures appears. And then what the colleagues did, they took this from 18 nestlings. They took, excuse me, they took cloacal samples from different nests, and then they sent these samples to me, to Vienna, and then I performed isolation and characterization of these bacteria, meaning in Enterobacteria, Enterococci, Staphylococci, and Mammaliococci. You see how Professor Miguel is taking a swab from nestling, and this is very nice small bird. And then what we found 44 different isolates. The predominant was E. coli, and the most important finding of, or more interesting finding of our study was that we found multi-drug resistance in 2 E. coli isolates, and in 1 isolate from Enterococci fitium. And what is also important to say that multi-drug resistant E. coli belongs to pandemic clone sequence type 58, which is which was found global in people, in humans, and in animals. And outlook of this study is that several isolates may be considered virulent, because we found also we could detect virulence associated genes in those isolates, which are important in development of pathology. And these isolates could be potentially pathogenic for animals and humans. Also, the growing evidence that antimicrobial resistant bacteria are currently part of the microbiome of wild animals, and this all underscores the need of one health approach. And the longitudinal study is needed, because to see if this phenomenon is just a transient colonization or persistent. And then I will switch now from the North America to Africa, especially to Rwanda. Why to Rwanda? Because I'm active there since 2015, and this is you see this red dot. This is a very small country in the eastern part, belongs to eastern part of Africa. It is bordering on the west with Congo, and the east with Tanzania, north with Uganda, and in south with Burundi. And this is the Rwanda. And if you see this black line, I put this on purpose. And I did this the last year for the students of a Western University. And I said here, take a look here on the right, on the eastern part. This is Pomona, and the end is in Santa Barbara. So, excuse me. So big is this country, meaning this is very small country, but highly populated. They have more than 14 million people there. And excuse me, how the whole situation began in late 2015. 3 colleagues from Rwanda pop up in our laboratory. Probably the colleague told me during the summer that someone from Africa will come, but I forgot, because a couple of days afterwards I had a very important presentation in Chicago, and I had to fly to Chicago. And then I didn't speak a lot with these people. 2 of them, these 2 persons, this is Beatrice and Jean-Claude. They were actually sent from Rwanda because they organized, my Austrian veterinary colleagues, they organized an NGO which is responsible for founding of a New Vision Veterinary Hospital in Rwanda. and then they acquired people, vets there, and these 3 persons were sent to Austria to learn. In the case of these 2 people, Beatrice and Jean-Claude, how to perform microbiology and parasitology in the laboratory, and the Vedaste, this was a 3rd person. He was more or less a clinician, so he wasn't in our laboratory just the first day. And after my conference in Chicago, I come back, and then my Africa story began. Until this moment, I didn't know anything about. Of course, I know where the Africa is. We learned it at school, and but I didn't have any association with safari or ideas I will fly to Africa or something like that. But in that moment, I started to speak with these people also after laboratory. And this was the rule that every single day I took them, and we go together after laboratory work. We go together to the city. We speak a lot. We speak about life. This was in the time Jean-Claude wanted to be married because he was in those days 30, and if you are 30 in Africa not married, you had some problem with your parents, in this case with his mother, because his father was murdered in this genocide in Rwanda. So, and I started to speak, and then I understood the problems of these people and how nice they are. And I also went with them to the museum. This is a natural history museum in Vienna. So this is a very famous museum. So we were everywhere together. Also, I'm doing swimming for all my life. And then this was a very nice story. The Jean-Claude come with me to swimming, but he couldn't swim, and I didn't know this. And he jumped in the swimming pool. This was the very, very, very nice situation. And so, but please remember this person. I will come back to him at the end of my presentation. And then they came after 6 months. They came back to Rwanda and started to work at a New Vision Veterinary Hospital, which was launched immediately after they come back. And what is the New Vision Veterinary Hospital? This is a center for research, education, and training for veterinary surgeons, veterinary technicians, specialists, and animal owners and farmers. And this looks like this. This is in northern province of Rwanda. If you are going from Kigali to Musanze, Musanze is this region. On the way from Kigali, which is the capital of Rwanda, you will pass this clinic. It is very nice and a big clinic, and this clinic has also laboratory capacity, and both Jean-Claude and Beatrice, but also my colleague Dr. Otto Fischer, who is the founder of this Rwanda story. They asked me to come to Rwanda, and then I decided in June 2017 to fly first time to Rwanda. And like always, I am doing training with students and the local population, local team. And how I do this workshop. First, we have a small theoretical part. You see, probably I gave a very complicated, I don't know what, to Beatrice. She is complaining here that it's difficult. And after theoretical part, we are going to the laboratory there to do bacteriology. And always I have students from University of Rwanda and the local team. And then what I'm doing, I have small groups so that every single person can learn, and every single person have to do bacteriology, to just that I'm doing, and one person and another person, and everyone is looking around. This is, I know this from my student time. This is nothing. You have to give work to people. And you know what I also achieved in Rwanda, that every time when we have a lot of fun in laboratory with people from Rwanda, and when I say, could you please sing, they are singing. So this achievement I don't have here in our Austria. I didn't have, we have also in Austria a couple of times a lot of students, but I didn't achieve with them that they are singing. So this is also very nice in Africa. And they are also we have a lot of jokes in the laboratory. It's a very nice atmosphere. And I was the second time also in December 2017, and then we start. We start. This whole story began much more intense, and in the meantime, we have memorandum of understanding between my university, University of Veterinary Medicine in Vienna, University of Rwanda, and the New Vision Veterinary Hospital. And all these three organizations organized in 2019 mastitis symposium. Why mastitis? Because you know, the cattle, especially cow, it is very important for the people of Rwanda. They have also this program, one cow per family. It is not only because of this, it is also connected with the history of these people. So this is, and you know, if these animals are sick, mainly through mastitis, this is also a very important issue. So we stay with this mastitis until the present. It is also part of our projects. And you see, during this project, I was here speaking with Letitia. In those days, she was dean of the veterinary college, and now very famous person in Rwanda. She's in Senate in Rwanda. By the way, Rwanda is one of the rare countries where you have in politics much more women than men. So probably we will see what that does it mean for the future. And here is our rector. And you know, on this picture we have a couple of students. You know, they have New Vision Veterinary Hospital is organizing every single year so-called castration parade. What does it mean? The 5 students from our university and 5 students from Rwanda, they are put together and they do castration and also vaccination together. And this is our team during this mastitis symposium. This is the founder of New Vision Veterinary Hospital. This is the Helga. She's in the meantime my doctoral students, the colleagues from Africa. Letitia, our rector. Bea is also a very good colleague from milk hygiene here at the university. And then during this symposium, the PhD idea was born, that we organize a student who will be doctoral students here in Vienna from Rwanda. Then we have a little bit break during the COVID pandemic, but we still managed to organize a lot of things in Rwanda, but also we invited Emmanuel, who is the lecturer at the University of Rwanda, and he told us that, and he was proposed by his university to come to Vienna and to see how it is working in Vienna, the climate, the laboratories, the possibilities. And afterwards, if he would be happy with this, to enroll for his doctoral studies. This is equivalent with PhD. And you see him working in our laboratory, and he's here with Dr. Otto Fischer, my colleague, who is the founder of New Vision Hospital. And then in 2022, I came back to Rwanda to establish PCR. This time I was not alone. I was with my colleague Damir from Croatia. He's also veterinarian, and he helped me to organize everything regarding PCR in Rwanda. And like always, we are starting with the theoretical part, then we go to the laboratory, and like I told you, every single person has to go from the isolation bacteria, isolation of DNA to the PCR, and we did this. And you know, this is also a very interesting picture when we didn't have this dark room for transillumination, and we organized something like this. And this is me And then I took a picture from our first PCR, which is a very good achievement in these conditions there in Rwanda. And then the last 2 days we went to northeastern part of Rwanda, which we have which is College of Veterinary Medicine situated, and here we discuss about doctoral thesis of Emmanuel. Here is Dean Martin and my colleague Damir from Croatia and myself. And the second day we have the symposium about, of course, my antimicrobial resistance. And here you can see another lady. She is Lydia. She is my second doctoral student, and here is Emmanuel. This is in the meantime my third doctoral student. So I have 3 doctoral students at the moment from Rwanda. And this lady, this in red one, this is a student, when I'm not mistaken, from this was an exchange student from Wisconsin Medicine University. And what we are doing in Rwanda, we are helping in improving veterinary microbiological diagnostic in Rwanda, and doing training and capacity building. And like I told you, bovine mastitis is very important issue in Rwanda, and we have here focus on staphylococci. But also my research interest in Rwanda is, and also from the whole group, is about antimicrobial resistant bacteria associated with companion animals and livestock, and especially in the last project with wildlife. And this is how it works. You can see a couple of pictures here during mastitis project. The students are taking the samples here and here. These are 2 Rwanda students in the laboratory at the New Vision Veterinary Hospital. This is Helga, my doctoral students. Agar plates, the first time we had Staphylococcus aureus, and when we have Staphylococcus aureus on our bacteria of interest, and we cannot perform further analysis, they are sending to Vienna. We do in-depth characterization, meaning multi-depth analysis and DNA microarray or full genome sequencing. And in the meantime, we have already 4 publications. 2 are about mastitis. About mastitis, and the last about was also somehow connected with mastitis, but was not with Staphylococcus aureus, but not mastitis. This was another colonization of ruminants in Rwanda. And very recently, our a manuscript about the diversity of extended spectrum cephalosporin-resistant Enterobacteria was accepted here for publication in BMC Veterinary Research. Our manuscript about this is the main topic of Lydia's dissertation or doctoral thesis, is about Staphylococcus aureus from dogs and cats in Rwanda. And we are now writing our manuscript about occurrence of MRSA and beta-lactam producing enterobacteria in domestic pigs in Rwanda. And then what we knew until now, that the antimicrobial resistance is a problem in Rwanda, both in human and veterinary medicine, and improving of veterinary microbiological diagnostic is needed in Rwanda. And from our experience, we know that we have some emerging Staphylococcus aureus clonal complexes, especially associated with bovine mastitis. This clonal complex 3666 we isolated for the first time in Rwanda, and we have a very new publication from Tanzania. So meaning these are the major countries affected with this Staphylococcus aureus. We don't know if it is new because nobody before us, nobody make this analysis, or it is really emerging. And what we saw also that we have presence of human-associated Staphylococcus aureus clonal complexes, minority in livestock, majority in companion animals. You will say, okay, this is logical because, you know, the proximity to humans, but I will come back in a minute to this issue. And then that some ruminant ESBLs belong to important pandemic clonal lineages. We have classical porcine situation, meaning we have ESBL and MRSA in pigs. And then the question is, what is about wildlife? And this is the reason why we organize our project. I will come back just in a minute to our wildlife project. And why I put this logical Because if you think like a Western person, you know, the dogs are in high proximity to humans. They are in rooms with you. They are in apartments, and you know this is not the situation which you will find in Rwanda. The majority of dogs are actually dogs for security purposes, and they are either on leash like this or are not. Sometimes they don't have any proximity to humans. But on the other hand, you see this picture I got from my colleagues from Rwanda. You will have the people. This is the mother and the one child, the second child. They are living in very close proximity to ruminants. So this is what we can know as a logical here in Europe, but I think also in the United States is sometimes you cannot apply this also to Rwanda. And what we understand that this One Health approach is extremely important, and that's why we organize also the project with wildlife. Why wildlife? Because Rwanda has a very unique wildlife. Just like for comparison, in Rwanda you will find, excuse me, you will find 221 mammal species. In comparison to United States, you have much more. But how bigger United States is in comparison to Rwanda? You have the population of these endangered mountain gorillas. Here, the Rwanda is home of big five, meaning elephants, lions, buffalo, rhinos, and leopard. And this, in the last couple of years, the wildlife plays a very important role in lives of the people of Rwanda through the tourism, safaris, and similar. And then we organize our project. It is about antimicrobial resistant patterns at the domestic wildlife interface in Rwanda. We have three regions. Remote, this is Akagera and Volcano National Park. Remote is the region where you don't have you don't have a lot of people, just a couple of tourists and rangers. The peri-urban region is the region with you have a little bit people, but not completely urbanized, and wild animals, and then you have urban centers. What we did in Akagera, we take a look on fecal samples of elephant. Why elephant? Because it belongs to this megafauna. It evoke emotion to people, especially children. Then we have African buffalo. We have a buffalo in both national parks, Volcano National Park and Akagera National Park. We take examples from zebra. On the beginning, we wanted to take examples from bark dog, but this was not possible. I couldn't believe this. You know, we saw a lot of these war dogs there, but couldn't find any fecal samples of them. Then we switched to hippo. And from monkeys, from primates, we have samples from baboons, blue monkey, vervet monkey, and you will find they're everywhere. So they are our sentinel species, because you will find in remote region, peri-urban and urban. And this is our remote region, Akagera National Park. This is on the border in eastern part of Rwanda, border with Tanzania. This is in comparison to Volcano. You see the completely different completely different climate. Here you will have in Volcano much more trees in comparison to Akagera, which is Eastern African savanna. And this is our peri-urban region, like I told you, a couple of houses, and then you have so-called nature with wild animals. The second picture, and then we have urban region. If you don't believe me, I have a couple of pictures here with monkeys. They are everywhere in urban region. This is Nyagatare city, which I told you the University of Rwanda is situated, especially a College of Veterinary Medicine, which is interesting for us. And then you will have monkeys everywhere. Going around it. Of course, they have this interconnection with people. And in the last year, I went there to see how much effort and workload we have to organize with this project. And this is my doctoral students, Emmanuel and myself. Why I put this car here? You know, if you are collecting samples and go to Akagera National Park, you have to go up and down 30 to 40 times during the day. We start 5 in the morning and until 2 or 3 o'clock. I'm in very good shape and I do sport for my whole life, but you will be exhausted on the end of the day. This is on the entrance of this national park, and you see the whole day we are speaking about feces, because we are isolating bacteria from fecal samples, and you see Emmanuel here is taking sample. This is our ranger. They take care about us. This is important because of security. And this is me taking samples. And this is also an interesting picture. If you have a small screen, you will not see, but here in black, these are buffaloes, and Emmanuel and one ranger go there to take fecal samples. Okay, this was not, they were not so close, but in this moment I was thinking just where the trees that I can go up, because there are some these buffaloes could be very dangerous. And here are both of rangers and Emmanuel, and you see if you are going there, you can come very close to zebras. This is the difference with if you are doing safari, you are not allowed to go around, and we were allowed because we performed research there. There you see the zebra. And then we took the samples, and what we did, we go back to our New Vision Veterinary Hospital. This is just a picture how Rwanda is looking, typical Thousand Hills. And you see, this is our clinics. 5 years. This is the picture from last year. You see the trees; they are very fast growing in Rwanda because you have sun and water. And then we start like every single workshop, first theoretical part, then we go to the laboratory and prepare our sample. And this is our students. He's performing, preparing agar plates. And you see, this is very interesting picture. All people are wearing masks. And this is this person, this student, and myself. We didn't, and you can say, Igor, you are ignorant because of pandemic. But the problem was not pandemic. The problem was this. These are our swabs in enrichment broth, and if you have fecal samples, the smell of feces is produced by bacteria, and if you enrich this bacteria, you will have a specific smell in the laboratory, and that's why the students wear this mask, and they told me, how can you survive with this smell and things? But I'm doing this for the last 20 years, and I used to have this smell around me in the laboratory. And this is our group of students from the last year, and what we expect from our wildlife study, that or in general from Rwanda, that the presence of human-associated bacteria and or antimicrobial resistant bacteria in domestic animals underscore the need for One Health approach. Our wildlife project will have a significant impact on environmental studies, public health, and conservation initiatives in Rwanda. You see here another picture I took the last year from buffalo. If you see buffalo like this is not a very, very nice situation, because they are very dangerous. And one day we would like to have involvement of medical doctors, because this interconnection in One Health approach between veterinarians and medical doctors is extremely important. But somehow it is difficult to interconnect. The same problem we have also in Austria here, I would like to say. And also awareness is our issue, that the people know how important this is, how important it is to send the samples to perform bacteriological examination and susceptibility testing. This is very important issue. And then finally, this is my almost last slide. I would like to show you come from One Health to One World. This is the family of my the doctoral students, Emmanuel. This is Damir, who went to Rwanda with me. And this is what I told you. Remember this guy? This is Jean-Claude from the first picture of my Africa story. Now with his family in front of his house. I always visit him and his family, and we are doing then. We are speaking a lot. We are eating together, and here I'm with the youngest child. And this, the last picture, what I would like to show you today, this is Igor, this is myself, but this is also Igor. So Jean-Claude named his son Igor because we have so good relationship and we stay in contact until the present. He's not at the New Vision Veterinary Hospital anymore. He's doing animal welfare, very famous now in Rwanda regarding animal welfare. But you know, this is a very also a success for me that you have a very small child in Rwanda called with my name. So this is you know, honor to what Jean-Claude did to me. And this is my last slide, and I come to my end of my presentation. And if you have any questions, and if we have a little bit of time, I would be glad to answer a question or more. So thank